right, guys, let's go ahead and get started here. I'm sure you guys don't want to be here any longer because you just got done with an exam, right? Okay, well, we've got a good news. It's a short lecture. But there's a lot of information in it, which is the unfortunate side of the lecture. But it's okay. We're going to be talking about selective and differential media, as well as another little lab that we're going to be doing on the results day as well. On Canvas, there should be a downloadable page that I think it says, it's basically kind of like a fill-in-the-blank review guide for this. It's not something that you turn in, it's just for you guys to study with and use, so don't feel like you have to turn that in or even do it. But if you do, it does tend to help people out. Okay, so we're going to be talking about a lot of different plates. For each of these plates, we've got two major criteria that you guys should know. Is selectivity and if it is differential. Most of the plates that we're going to be talking about are either selective and differential or just selective or just differential. So what does selection mean? It means I only allow a certain type of thing to grow on me. I don't allow anything else to grow on me, just this one particular type of organism. So that's what selectivity is. Differentiation gives you information about the bacteria that are growing on the media. So the bacteria have to be growing in order to show you to show you information about that thing. So differentiation is on bacteria that are growing on that plate. Selectivity is whether or not bacteria were able to grow on that plate or not. So the first plate we're going to talk about is mannitol salt agar. As the name implies, it's got a lot of mannitol and salt on it. It is a bright red media, so it's super bright red. It's kind of see-through, so you can look through it and see light come through it. It has a pH indicator in there called phenol red, which we'll use a lot of in lab. The sugar in here is mannitol, and it contains about 7.5% salt, which doesn't seem like a lot, but it's actually a huge amount of salt, and it's really only a few types of organisms are able to survive on it. So specifically, this type of media selects for gram-positive halophiles, or halotolerance is another word for it, organisms that can withstand really high salt concentrations. Some organisms really like and thrive under those conditions. Other organisms can just tolerate it really well. So anything that can handle that, we'd say is a gram-positive halophile or halotolerant organism. Now, this media differentiates the ability to ferment lactose. And you guys remember, ferment mannitol, not lactose. This one's mannitol. When you ferment a sugar, you tend to get a couple of different byproducts, but it generally falls into three things, which is an acid byproduct, CO2, right, and some sort of alcohol. Not always, but a lot of times you'll get an alcohol. And so fermentation process, we can really easily test if the pH changes or if CO2 is produced. So in this case, we're looking for a pH change. So phenol red is the pH indicator here, and it'll change to a nice bright yellow if it gets very acidic. So the more acids being produced by bacteria, the more, that means that mannitol is being fermented and it's creating that nice yellow color. So this is what it looks like on a plate. So the plate itself is normally this bright red. Right here you can see a string of bacteria growing on here, and the bacteria are growing, but the plate is still red all over. So that means that this bacteria is a gram-positive halophile, but it is not a mannitol fermenter because it's not changing the color of the media at all. The guy right next to it, though, this one's growing on here, and it's created that nice bright yellow coloration. So if it's got that bright coloration, that means it's producing acids because it's fermenting. That means it is fermenting mannitol. So we'd say this is mannitol positive. This organism can metabolize mannitol. So pretty easy one, not too complicated, very, very simple. And it is one of the easier plates to read. Now, the next plate is called McConkey's agar plate. It is also selective and differential. This one's still kind of red. It's more of a violet color, so it's kind of like this faded red color. The sugar in here, though, is different. It's lactose. And it's got a couple of other special things that's been added to it. It has these bile salts and crystal violet. If you guys remember crystal violet, that's what we use for gram staining. And these prohibit the growth of gram-positive organisms. So it basically says, hey, if you're gram-positive, you cannot grow on this plate. So it selects for gram-negatives. It also has a pH indicator in there called neutral red, and neutral red will help us monitor any kind of production of acid from this bacteria. So it selects for gram-negatives, and it differentiates those that can ferment lactose. There's 3 categories for lactose fermentation. So you can be a non-fermenter, you just don't ferment anything, and if you do this, the way you grow on this bacteria, bacteria grow on this plate, they'll just be normal bacteria color. They'll be kind of like this translucent white color. They won't really have anything to them. But if you're a weak fermenter, so that means you can only ferment it a little bit, you're not very strong at it, you're only going to produce a little bit of acid, so the bacteria themselves will change color to this kind of slight pink coloration of colonies. But if you're a strong fermenter, you will turn into these hot pink colonies, and then you will change the color of the agar around you, creating this halo effect where you're pulling in the bile salts and you're changing the color of the agar to hot pink. I'll show you guys an example of that. So here you can see the plate, so you can see it's kind of like this faded color. When it's uninoculated, it's a little bit darker red than this, so just keep in mind. Now this bacteria is growing on here, so that means that it has to be a gram-negative. You can see it's kind of got this dark purple coloration to it. So most bacteria are kind of this purple pinky color. So this would be a lactose fermenter, a weak lactose fermenter. This guy you can see growing on here, you can see the bacteria themselves are hot pink and have created that halo effect where they're sucking in the salts around them and creating this nice hot pink look to them, halo effect. So that would be a strong lactose fermenter. Here you guys can see this one has bacteria growing on here, and they are gram-negative because they're growing on here, but they don't have any coloration to them at all. So they are non-fermenters. So they cannot ferment lactose at all. Now, you can see that some people have done some really nice, clever little art with these, which we'll talk more about in just a second. But they've done some really cool pictures. I stole these from the internet because I don't have any artistic talent to do this, but just be aware that you can do some kind of cool things with it. Now, the biggest issue, though, is if you have things really close together, we do not want to do little zigzag lines when we do these kinds of inoculations. We just want to do one straight line, because you can see like right here, this one's a strong fermenter, but it's really hard to see the halo here because you've done the zigzag, and the zigzag kind of distorts that halo effect. So just do one little line. You don't have to do a giant line. You don't have to do zigzags or anything like that. Just one tiny line, okay? All right. The next plate is EMB. EMB is a dark red, maroon-looking plate. It has a couple of interesting facts to it. First of all, the sugar on there is lactose. And EMB stands for eosine methylene blue, which are the chemicals in there, eosine Y and methylene blue, which are types of stains. And they will inhibit growth of some gram-positives. So it tends to select for gram-negatives. Now, if you notice, the sugar in here is lactose, so that means we're looking for lactose fermentation. And if you're really, really astute, you might go, hey Josh, why are we doing 2 plates that do exactly the same things? Which I would say, good question. The reason why is because each of these plates have different strengths. If you notice, this one says it only inhibits the growth of some gram-positives, and the other plate that we were looking at, the McConkey's, prohibits, so it doesn't allow any growth of gram-positives. So the other plate's really, really good at selectivity. But this plate, you'll see in just a second, the differentiation for lactose is a lot clearer. So this one, a non-fermenter, is just translucent. The bacteria don't have any color. You can just see right through them. There's nothing special about them. The ones that do weak fermentation will have dark maroon colored colonies on them. So they'll kind of take up that red looking dye, and there's going to be this dark red look to them. If you are a strong fermenter, though, you will create a metallic green sheen. So you'll shimmer when you hold it up to the light, and it'll kind of reflect the light like a metallic foil, which is really, really easy to see. So EMB, much, much easier to determine differentiation on it, even though it's not as good as selectivity. McConkey's way better at selectivity, but not as clear for differentiation. It can be sometimes obscured. So this is an example. Here we've got E. coli, and it's got this nice, brilliant green sheen. So it's gram-negative. It likes to grow on there. And it's a strong lactose fermenter, so it creates that brilliant green sheen. Here we have a plate with 4 different organisms on here. In this area, this organism is actually a gram-positive, so it doesn't grow on here. This organism is a gram-negative, but it's an oxidative organism. It does not ferment sugars, so it cannot process lactose, so it just doesn't have any coloration to it. It's just translucent bacteria. The one right here, though, has this dark maroon coloration. It's a little hard to see on the lights there. I'm going to turn the lights down just a second. There we go. That's much easier to see. Okay, so it's got that dark maroon coloration. It's very dark. That's a weak fermenter, and the one on the other side has that metallic green sheen. So it's really bright and brilliant. It's super easy to see. So those two plates have overlapping functions, but one does one thing better and the other does the other thing better. Okay, the next plate we're going to talk about is blood auger plates. Blood auger plates, a little bit tricky in that you look at it and it's the same color as an MSA plate. But the difference is that the blood auger is completely opaque. So if you hold it up to the light, you can't see anything through it. You put your hand behind it and it's just completely opaque. MSA, you hold it up, you can see your hand through it. It's very translucent. Now, blood auger, as the name implies, has blood in it, about 5%, typically sheep's blood. And we use it because it is actually really good at allowing bacteria to grow on it, especially bacteria that are very picky at what conditions they have. We call them fastidious organisms. They require specific nutrients and conditions to grow properly. A great example of this is Streptococcus pyogenes. It can grow on TSA, it just doesn't like to. But if you put it on blood auger, it grows really well, no problems. So, this organism, or this plate, isn't selective. It is actually only differential because it's just nutritious. So it allows anything to grow on it. So it's not a selective plate, but it is a differential plate, and it differentiates between hemolytic states. If you're alpha hemolytic, that means you can partially lyse some red blood cells, so you're not doing a great job of it, and you're going to create this dark brown coloration on the colonies themselves and around the colonies. If you're beta hemolytic, that means you can fully lyse all the cells and create a clear zone. So you can actually see through the plate now because it's eaten away at all the red blood cells, and now it's completely translucent in the media. The last one is gamma hemolytic. And gamma hemolytic is actually kind of a trick word. It's not hemolytic at all. It is non-hemolytic. So that's another way we refer to it. Basically, the bacteria is just growing on the plate, but it doesn't have any change of coloration. It's just normal colonies on here. So you can see that they haven't changed the color of the auger at all. They don't have any discoloration to them or translucence. So that means that this is just a gamma hemolytic or non-hemolytic bacteria. Either way is fine, however you want to say it. So this plate only does differentiation. It doesn't do any selection. If we look on PEA auger, it's an interesting auger because it uses this chemical called phenylethyl alcohol, which is actually a very bizarre kind of oily alcohol. And it mostly selects for the growth of gram-positives. So it basically doesn't let any gram-negatives, not many, grow on there. It kind of just inhibits their growth, so it reduces the amount of growth on here. And if you look here, plate A is a PEA plate, and you can see the gram-positive organism growing here very, very robustly. The gram-negative, though, it's still kind of on here, but it's really suppressed. So if you start seeing like the bacteria like struggling to survive on there and not growing very well, we would consider that a gram-negative. It's pretty easy to differentiate it that way. Here's a TSA plate. You can see both on the TSA plate, because it doesn't have any selective or differential properties, they both grow on here really robustly, but on the PEA plate, the gram-negative is really struggling to survive. So this is a selective media only. It doesn't differentiate anything. So, fun little fact, because it is really good at selecting, we'll sometimes combine it with the blood auger, so it becomes a selective and differential plate. But we won't be doing that. We're separating them out. And the reason why we do that, a lot of times if we're looking for specific types of organisms, we just want the gram-positive organisms, especially for like diseases. So we would want to basically eliminate any gram-negatives growing on there, so we'd add PEA to it to make it a selective plate and then differentiate the bacteria growing on there as either alpha or beta hemolytic or non-hemolytic. Okay, so that's those plates. Now, what you guys will do is you guys will get 6 different types of plates, 2 sets per group. So each pair of lab partners will have these 6 plates. And if you guys notice, I said 6 plates. We'll talk about the last plate here in just a second. You guys are going to, of course, label them, name them. And a really key thing on here is making sure you jot somewhere on there what type of media that is, just so you don't get confused later on. It is pretty easy to differentiate them even after they've been inoculated and incubated for a while, but sometimes it's hard if you haven't done it before. So you might want to just jot on the side of the plate, hey, this is MSA, this is EMB, what have you. You'll take a sterile inoculating loop, and like we've done in the past, you guys will streak them. Now, this one is different because we're not doing streak for isolation, we're just doing a little line there. And the way you guys do the line, is a little bit different than the picture because obviously I wanted these to be readable. But the line will just kind of be across here, and then up and down here, and up and down here. And the reason why we do that is it helps keep the maximum distance between the organisms so that we don't have any crosstalk between them or difficulty differentiating which ones are going which way. So make sure you're very careful with inoculations. Also, if you notice on here, each plate has its own set of bacteria for each inoculation. Like here, we have E. coli growing on all 3 of these plates, but there is no E. coli growing on the blood agar. So we don't want to do the inoculations willy-nilly. You want to make sure you're very, very specific. I tried to color code them so that it's easier to see. So E. coli is being put on MacConkey's, EMB, and MSA, but for example, Enterobacter origines is only on MacConkey's and EMB and not on mannitol salt agar. This one's Enterococcus faecalis. So very, very carefully make sure you guys inoculate them and make sure that you have enough space between them. Once you guys do all of your inoculations, it's really important to sterilize your loop in between inoculations. You will then tape all the plates together in a stack, with the exception of blood agar and PEA plates. You guys will tape those together, and you give them to your TA because they will put them in a Ziploc bag that's sealed with a little CO2 pouch. And that's because blood agar and PEA organisms do really, really well when they are in a predominantly CO2 environment. So it's just kind of an easy way to do that is put them in a Ziploc bag with a CO2 packet in it. So make sure you guys keep that in mind. And then you guys will incubate them for at 37 degrees Celsius for 48 hours, and then you guys will read the results. Really easy to read the results, like we talked about before. If you guys do get stuck on something, make sure you ask your TA or your LLAs there for help. Now the last plate is Mueller-Hinton, which we haven't talked about yet. It is just a regular old nutritious plate. It's used typically for doing antibiotic growths because of the way it kind of has a very stable way of having bacteria produce certain enzymes. And so we tend to rely on this for certain types of things, but it's basically just like TSA plate, only it's made with Mueller-Hinton agar. And we have 4 organisms on here, and we are going to be using these organisms to do some of that agar art we were talking about before. So we have 4 organisms. One we used last week, Pseudomonas aeruginosa, which is produces kind of this green pigmentation on it. Serratia marcescens, which you guys have seen before, that makes that nice bright red coloration on it. Micrococcus luteus, which is a bright yellow bacteria. And lastly, Cochuria rosea, which is this bright pink coloration colonies. And I know it says on here, standard quantitative analysis of water. We aren't doing that, so just ignore that. You don't have to pay attention to that at all. But we will be doing a bioart thing. And so we were showing you guys pictures before about the bioart. For this one, art's actually been a very important part of science. Back in the day, you actually kind of really had to be an artist because you had to sketch and demonstrate your submissions in your books or to journals of things that you observed. It'd be terrible for me because I'm terrible at drawing. And so, I don't have the talent of like this scientist right here who drew this woodpecker freehand while they were out in the woods. Like that's what they did out in the woods while they were watching them. But it's still kind of carried over, and a lot of scientists still use art a lot, especially in microbiology, and they will use the bacteria and fungi and other organisms that we have available, as well as the media that we use to create different types of art. And we even have competitions. So this one's actually the winner of the National Geographic Science Competition for agar art from, I think, 2018. And they used a mold to create this giant volcanic mound, and some other very basic bacteria, as well as, I believe this is Serratia that's creating this red here. And they use a special type of media that has a nice blue look to it. So that's an example there. And another guy, he created this really interesting way. This is also bacteria. This is Serratia marcescens, and the way he did this is he created an even distribution of Serratia, so he suspended it in liquid, put it on a plate, spread it across evenly, and then he took a negative film, put it over the top of the plate, and then he shot through the film a high-intensity radiation, I believe it was X-rays, which would kill off the bacteria. So anywhere on the film where it was dark, it would block the radiation and the bacteria would survive. But anywhere on the film where there is light, it would shoot through the radiation and it would kill the bacteria underneath, and so you'd create these prints of bacteria on these plates. It's actually a really cool thing. So he did this one with Albert Einstein. He's done a bunch of others, and it kind of creates that point-related kind of old print style picture. So it's actually really, really neat. Little bit of art that he's done there. We've all done this in the past for students. So these are some things that students have created. They used different types of media. So here somebody just used TSA to make these nice little hearts, as well as this flower, which is actually 3 different types of bacteria. It's actually really, really hard to do this, to get it right. So they actually did an amazing job on this one. This is one of our actual learning lab assistants in lab. She made a little baby Yoda, which is great. And then we had some students who had done a special research lab to develop and design different methods and techniques to help us make more clear auger art images, which I'll share with you their results in just a second here. But these are some of the images that they created. So they got these very fine lines made. It's really neat how they did it. Now, what you guys will do, you will have access to any of the bacteria that you guys made from the week before, or from that week. So on the results day, which for you guys would be Thursday, you guys will inoculate either a TSA plate or any other of the other types of plates that we have available, and make a picture of it with some sort of art using bacteria that we have access to. We will also have a couple of other unique organisms that you guys can use that we don't have included in the inoculations that we're going to be doing. So those ones are a little bit special. The inoculation will happen on Thursday. We will view the results the week after that on Monday. For you guys, it would be Tuesday. And we will actually vote on which section's auger art thing is their favorite, and that person will win a small prize. Now, the way you guys can do this, okay, very, very easy to do. They've created, the students that were helping us before, created kind of a guide of different types of coloration, as well as different ways you could manipulate the bacteria, and they kind of have a highlight of different ways in which you can do it. One of the ones that I thought was actually really, really interesting is they really pointed out things that require like spacing. So if you have too much growth spacing-wise, or too much stuff in a space, it will kind of overlap and make it very difficult to see. And then they also showed the difference in temperature growth. So if you grow at a very hot temperature, you're going to get a different kind of pattern than if you grow it at a much lower temperature. And the last thing that they did to get these like really, really, really fine patterns on here, which I thought was really interesting, is they would draw on a sheet of paper or on the back of a Petri dish the design that they wanted to do first with a pen. Then they'd flip it around and copy that design with a needle, doing it very, very finely to get it to make it look nice. Now, doing it with a needle takes a lot of talent and technique because the likelihood of you gouging the auger is really, really high. They spend a lot of time kind of developing that technique. But you could do it also with a loop, but that's a kind of a little trick, is to do the drawing pattern and put it on the back of the plate, or draw it on the back of the plate first, and then draw the bacteria on the auger. So really, really cool little set. There's this page is up for you guys to look through, so you guys can look at some techniques if you guys are really curious in doing it and maximizing it. Okay, does anybody have any questions over anything that we've covered? Everything make sense? Yes, good. All right, that's it. You guys are done. Easy, quick thing. Have a great day, and I will see you guys next week. And if you have any questions, come up and see me.